welcome to our channel. In this video, we are going to discuss about the enzyme catalysis and their types. So, enzyme catalysis is the formation of the binding energy, and the binding energy is the energy which is formed by the interaction of the substrate and the enzymes. And this free energy helps to lowers down the activation energy. So, there are three types of catalysis, that is acid-base catalysis, covalent catalysis, and the metal ion catalysis. Acid-base catalysis involves specific and the general acid-base catalysis. Specific catalysis is a type of catalysis that uses only the hydronium ions and the hydroxide ion present in water is referred as specific acid-base catalysis. And general acid-base catalysis uses the weak acid and the weak base other than the water. So amino acid residues, general acidic form and the general basic form. So for glutamic acid and aspirin, RCOOH carboxylic acid is present and the RCO minus. And for lysine, arginine, cysteine, histidine and serine tyrosine are given as follows. So in this figure, the hydroxyl group, which has a positive charge ion, is interacted with the negative ion charge, and substrate 1 is there which interact with the enzyme, and substrate 2 is not interact with the enzyme. So in general, specificity is derived from the formation of many weak interaction between the enzymes and its specific substrate molecule. So binding energy, and it's the major source for free energy used by enzyme to lower the activation energy for the reaction. So next one is the covalent catalysis, in which the reversible covalent bond between the enzymes and the substrate, and these interactions are temporary. Enzyme performing covalent catalysis be nucleophilic and the electrophilic centers. Example, chymotrypsin. It acts as covalent as well as acid-base catalysis. The third one is the metal ion catalysis, in which also called as the Lewis acid-base catalysis. So ionic interaction between an enzyme bound metal, that is Mg plus, and a substrate CO2 can help and orient this this substrate for the reaction or stabilize charge reaction transition state. So carboxylated side chain CO2 plus ribulose 1,5 biphosphate. So in this, if no enzyme present, then free energy, that is Gibbs free energy, increases. And if the difference in the free energy between the transition state and the ground state is called as the Gibbs free energy of the activation or the activation energy. So if there is no enzyme, then del G is increases, and enzymes complementary to the substrate, then diffuse products formation, and del G not del G is an uncatalyzed form. And if the enzyme is complementary to this transition state, then del G is increases uncatalyzed form and the reaction goes on. So enzymes reduce activation energy by due to the binding energy that is covalent interaction between the enzyme and the substrate. The maximum binding energy is released when the enzyme facilitate the formation of the transition state, the non-covalent interaction between the enzyme and the substrate. That's all about the enzyme catalysis and their types. Keep watching. Thank you.